Hi, my name is Sara Guerreiro, and I am a last year PhD candidate working in neurodevelopmental diseases at ICVS in Braga, Portugal. My research focuses on investigating the excess of neuronal inhibition in a brain region that is essential for learning and cognition, called cortex, in Down syndrome. More specifically, I am trying to clarify whether cortical inhibitory neurons production, differentiation, and migration are altered in Down syndrome, and how this is linked to the cognitive deficits found in this condition. The goal is to clarify the mechanisms mediating some clinical manifestations of this trisomy while identifying potential therapeutic targets to develop more effective therapies. To address my scientific question, I combined the use of human postmortem tissue with cell culture, using patient-derived human-induced pluripotent stem cells and the Down syndrome mouse model. Each method answers different parts of my question, providing complementary information. Using the human postmortem tissue, I can identify genes involved in inhibitory neurons development that are altered in Down syndrome, while in parallel, producing inhibitory neurons from patient-derived human-induced pluripotent stem cells allows me to tackle each step of the development of this neuronal population, from its production to its migration to its differentiation, giving me information about cellular and molecular mechanisms that may be altered. Finally, using the Down syndrome mouse model, I can study the involvement of these neurodevelopmental changes on the different behavioral phenotypes, hopefully contributing to the identification of more targeted therapeutic strategies. The most appropriate research model is selected based on the specific scientific question we are trying to answer. While human-derived cell models can be great to tackle human-specific cellular and molecular processes like disease mechanisms, drug responses, toxicity testing, or genetic manipulation, they lack the complexity of a living organism. So, if the objective is to study complex processes or to understand the impact of those in behavioral phenotypes, using in parallel both human and animal models improve both the mechanistic understanding as well as the translatability to humans. However, it is important to have in mind that when using animal models, we should follow the principles of reduction, refinement, and replacement to ensure responsible research design. By using new approach methodologies in my research, I can more easily dissect different developmental processes while precisely modulating genes or proteins of interest for a detailed investigation of cellular and molecular mechanisms that may be altered in Down syndrome. The initial approach refines my hypothesis and leads to a more targeted planning of the mouse experiments, allowing a reduction in the number of animals needed. Also, by combining both, I improve the quality of my data as well as the translational potential, increasing the relevance of my findings for the clinical manifestations of Down syndrome.